Hi everybody, in the previous video we talked about the mechanical properties of the hydrogels, and we mentioned that upon the injection of these materials, when the temperature is increased, we will have the instantaneous formation of the hydrogels. And this gelation is called the physical gelation, and it is quick because the bonds which form the network of the polymers are the physical bonds. On the other hand, if the polymers in this network are connected with each other with the chemical bonds, we will have the chemical linking, which is stronger, but it takes more time compared to the physical gelation to be formed. And for the physical gelation, because it is happening quickly and instantaneously, we can exploit this phenomenon in order to form the so-called microbeads or the tubes by dropping our hydrogel in the liquid form into the bath of hot water. You see here we have the hot water, and when we inject or we drop our material, the microbeads or the tubes will be formed. For the formation of the tubes, we can use these so-called molds, so they will help us to finally lead to the formation of the tube form of the hydrogels. And you see here in this diagram that the amount of elongation, the strain, how long our material can be elongated compared to the stress that is applied at the two ends of it, is a lot. So these materials can elongate a lot before they, let's say, they break, and they have their own applications based on the place or the type of healing that we are going to do. But the more important application of the, let's say, another important application of the hydrogels is for the cell microencapsulation. For example, you see here we have the matrix and the cells which have been implanted, let's say, have been encapsulated into this matrix. And what is the application of this cell microencapsulation? Just consider the time that the patient is suffering from diabetes. This patient needs to be injected by insulin every day, and the everyday injection of the, say, insulin is something problematic and causes some problems. So what is the problem of such patients? The problem is that the pancreatic islet cells are not performing well. So if we extract some of these cells from, for example, from an animal source, and we implant it into the body of the patient, they can start to, let's say, form and to produce the insulin in the patient again, and the problem of the daily injections will be solved. But the problem is that because they have the animal source, when they are implanted into the body, the immune system of the body will reject them. So this is the problem, and we have to provide an environment for them to avoid their removal by the immune system from the body. And this problem can be solved by the encapsulation of these pancreatic islet cells inside the hydrogel, inside this material. And the properties of this hydrogel is specific. Let me show you in this slide. As you see here, the hydrogel, the matrix in which the cell has been implanted, has been encapsulated, is a semi-permeable. You see here that for the small materials, for the nutrients which are essential for the cells, they can easily diffuse inside the matrix, and the therapeutic products and the waste products then can diffuse out of this semi-permeable matrix. On the other hand, for the antibodies and the inflammatory cells of the immune system, which are going to remove these cells out of the patient's body, because they are large, they will not be able to diffuse inside our matrix, and these cells can continue for the production of the insulin, and luckily the insulin is much smaller compared to the size of these antibodies and inflammatory cells, so the therapeutic products can easily diffuse out. But what we mentioned was just one example of the cell encapsulation for the diabetes treatment. We can do it for the so-called hepatocyte cell transplantation, and also for the treating of the cancerous tumors. Okay, let's go to the next slide. Well, I see that I have talked for about 6 minutes, and I will talk about the calcium alginate in the next video.